Now, let's turn our focus to something more realistic, the actual structures as they appear in real life. In other words, we'll be looking at photomicrographs, so we can observe and identify these leaf structures more clearly and see how they appear beyond the diagrams. So, here is our first photomicrograph showing a transverse section of a dicotyledonous leaf. Unlike the diagrams we just explored, this is an actual image taken through a microscope, giving us a real-world view of the internal leaf structure. When you look at it, you can see how the different tissues actually appear under the microscope. Right at the center, you'll notice the vascular bundle. This is the leaf's main transport system, often referred to as the central vein. In fact, in this case, it's specifically called the midrib, which is the large central vascular bundle running through the leaf. Around the edges of the leaf, you can clearly see the epidermis, which forms the protective outer layer, both on the top and bottom surfaces. Just beneath the upper epidermis, you'll spot a layer of tightly packed cells. While it might not be labeled in the image, this is easily recognizable as the palisade mesophyll, the main site of photosynthesis. Then, as you look further down, you'll see the spongy mesophyll. These cells are more loosely arranged and have noticeable air spaces between them, which help with gas exchange, exactly as we saw in the previous diagrams. Again, here, you can see both the diagram and the photomicrograph side by side. And you can easily identify the cuticle, which is a thin, waxy outer layer that helps reduce water loss. In the diagram, it's clearly labeled as a smooth line above the upper surface. In the photomicrograph, it may appear faint, but it's visible as a thin border over the top layer of cells. Just beneath that, we have the upper epidermis. In the diagram, it's shown as a single layer of neatly packed cells. In the photomicrograph, this same layer is visible and forms the upper boundary of the leaf structure, though the individual cells may not be as sharply defined. Next is the palisade mesophyll, a key site for photosynthesis. In the diagram, you can see long, rectangular cells stacked closely together just under the upper epidermis, each containing small green dots representing chloroplasts. In the photomicrograph, this layer is also clearly distinguishable as a band of elongated, tightly packed cells, slightly darker due to the presence of chloroplasts and staining. Below that, both images show the spongy mesophyll. In the diagram, these cells are shown as rounder and more loosely arranged, with large air spaces between them. You'll see the same thing in the photomicrograph, irregularly shaped cells with clear gaps in between, allowing for gas exchange within the leaf. Moving to the lower surface, the lower epidermis is shown as the bottommost layer of cells in both images. In the diagram, it's labeled and more neatly illustrated. In the photomicrograph, the layer is slightly less defined, but still forms a clear boundary. Finally, look closely at the guard cells and stomata, mainly visible in the diagram near the lower epidermis. These control the movement of gases in and out of the leaf. While in the photomicrograph they may not be as easy to identify at first glance, their position on the lower epidermis hints at where the stomata would be. So, we can observe that the diagram gives a clear, labeled structure to learn from, while the photomicrograph confirms what these tissues look like in reality. And by comparing both of them helps reinforce not only where each structure is, but also how they appear when you actually observe a leaf under the microscope. Now let's look at another photomicrograph. This one zooms out a bit to give you a more complete view of the entire leaf cross-section, from the top layer all the way to the bottom. And, keep in mind, this is an exam-style question taken from a past paper. Make sure you learn all the labels thoroughly so you don't lose any marks. At the very top, you can spot the waxy cuticle, again, a thin barrier that prevents water from evaporating too quickly. Right underneath that is the upper epidermis, a layer of protective cells. Just below the epidermis lies the palisade mesophyll. These cells are long, stacked closely together, and packed with chloroplasts. This is where most of the light absorption happens. Further down, you'll see the spongy mesophyll. The cells here are rounder and more spaced apart, with clearly visible air gaps that allow gases to move freely between the cells. Within the middle of the mesophyll region, a vascular bundle is clearly visible. This is the leaf's transport network. And if you look closely at the lower side, you'll see stomata, the tiny pores on the surface, along with guard cells that regulate their opening. These are found within the lower epidermis, which lines the bottom surface of the leaf. This image does a great job of showing all the internal components of the leaf in one view, and it really helps connect the structure of the leaf to its function in gas exchange and photosynthesis. Finally, we have this last photomicrograph which is also from your past papers, and this one takes us deeper to its structure, as it zooms in on a vascular bundle, which you can think of as the leaf's internal highway system. In this close-up image, you'll clearly see the vascular bundle structure. The xylem cells are the larger ones, usually toward the center. The job is to transport water from the roots up into the leaf, which is essential for photosynthesis. Surrounding the xylem are the phloem cells, smaller and more compact. These transport the sugars made during photosynthesis from the leaf to the rest of the plant, where they're either used for energy or stored. This zoomed-in view is important because it highlights just how critical these transport tissues are. Without water being delivered by the xylem or sugars being carried away by the phloem, photosynthesis wouldn't be possible or useful. So, with these 4 photomicrographs, we've gone from an overall view of the leaf, to a detailed cross-section, and finally to the fine structure inside the vascular bundle. Each one helps reinforce how every part of the leaf, its layers, cells, and tissues, is perfectly adapted to support photosynthesis, gas exchange, and transport, the key functions that keep the plant alive and growing.